talk about the cell today again, but much more specific. So we're going to talk about the part of the cell, which is the cell membrane. Why cell membrane requires a separate topic? Because, you know, it has a very profound effect on the cell structure, both cell structure and function. Membrane is the one that controls transportation. It also keeps the cellular contents together. It provides protection. And just to remind you, cell membrane is partially hydrophilic, partially hydrophobic, which actually allows to provide internal stability, the homeostasis inside the cell, okay? It's like, you know, a place where you live, right? This is your space versus whatever's outside, right? So the walls, the door, the windows are part of what encloses you and protects you inside your home, right? So membrane is pretty much the same thing, right? But your home is for you. The cell membrane is for the cells that make you up, okay? I can put it this way. So one more time, do you remember how many cells make up your body?
70 trillion?
75 trillion cells.
75, sorry.
Yes, and just remember also, on top of your own cells, you have bacterial cells as well, right? So in this picture, you see two cells membrane, right? Like two cells connected together. There are structures, remember, that allow this. Tight junctions, remember, desmosomes, gap junctions. You see the transportation vesicles loaded with something. And you can also see here, it's hard to say whether it's exocytosis or endocytosis, either picking up or releasing. Okay, and so membrane, as I said, is semi-permeable. Semi-permeable, which means that for some substances, it's kind of like, you know, they can cross the cell membrane the way they are. For majority, I would say it's not like that. For majority substances, they have to be moved through the cell membrane, in or out. What's the catch here? Do you remember molecules that are polar? What does it mean, a polar molecule?
That part of it is negative and part of it is positive, right?
Awesome.
You're
Fantastic, yes. Polar molecule means it has a charge, right? It has a charge on it. So maybe one region is positive, another region of the molecule is negative, right? But it has a charge. Non-polar means they have no charge. So non-polar usually move through the membrane. Wherever has a charge, or if the molecule is way too large to cross the membrane the way it is, it has to be transported. So transport through the cell membrane is usually divided into passive and active. Passive transport means no energy is required. So cell doesn't spend energy molecule, which is ATP molecule, to move substances through, in and out. Okay, so this is passive transport, you see. Some molecules can cross the membrane the way it is. As I said, this molecule should be non-polar. All molecules have to be moved through the transport proteins. And there is an active transport. Active transport means cell has to spend energy to move, right? To move things. And this is a pump. We're going to talk about this. This is a pump, which means that, yes, certain molecules, energy will be applied. And this is called bulk transport. Bulk transport when it's a big load, big delivery, or big pickup, right? So exocytosis cell releases molecules outside. Endocytosis cell picks up large molecules inside. Cyto means cell, exo means outside, endo means inside. And one more time, remember vesicle, right? So both exocytosis and endocytosis, they require transportation vesicles. And let's talk about the structure, right? So I think I told you that membrane is not, it's not a rigid, static, non-moving structure. Membrane is very much flexible, right? It's flexible, and flexibility is achieved by molecules that make up the membrane. So the core of the cell membrane is made of molecules called phospholipids. Phospholipids have two parts: phosphate head, which is hydrophilic, it interacts with the water, and fatty acid tails, which are hydrophobic, they repel water. So as it's stated in your slide, phospholipids are amphiphilic. I think I also explained to you a while ago what does it mean, amphiphilic, which means that they both hydrophilic and hydrophobic. So phospholipids, you can just break it down. You can say hydrophilic has hydrophobic tails, or you can say amphiphilic. It has hydrophilic region and hydrophobic region. So the arrangement of the molecules of the phospholipids is that the heads face outside and inside, okay? Let me just write something for you. A little bit easier. So if I draw this layer, let's say this is the part of the cell membrane. So this arrangement, having two layers of phospholipids, is called phospholipid bilayer. Bi means double. Okay, so phospholipid bilayer. So this, let's say, extracellular fluid, right? Extra, outside the cell. And this is intracellular. Intracellular means inside the cell, okay? So both extracellular and intracellular fluid is mostly made of water, right? Which means that water is a polar molecule, that these heads can interact with the water and whatever is mixed with the water. So they can interact on the extracellular side, they can interact on intracellular side, right? However, this water can't cross the hydrophobic tails, right? So they are hydrophobic. Means again, water as much as you want, whatever is mixed with the water, but the water can't cross cell membrane the way it is, okay? There is no way this water will be repelled by fatty acids that are hydrophobic. Is it clear, guys?
I have a question.
Please, Jamarius, can you turn the camera? I haven't seen you for such a long time.
Not right now, sadly.
No, what happened?
I just have.
Okay, bad hair. Okay, that happens.
Not even that, but okay. I was gonna ask what's between like the tails. Like, are they connecting with each other? Are they touching each other, or is there like a liquid or something in it?
It's a very good question. I'm getting to that right now.
Okay, sorry.
Okay, but what I want you to pay attention is that you see this fatty acids, they like one acid is saturated, which means it's straight, right? And another fatty acid is unsaturated, which means it has a kink, right? So one is straight, one has a kink, which means that they are not tightly packed, right? So there's a space between their molecules. And it means, again, like this one is kind of like pushing whatever's next. So creating this space. I don't see the picture here, but I think there will be, yeah. So yes, basically this is the answer to your question, cholesterol, right? But we'll get to this. So between the phospholipids, the fatty acids, there are cholesterol molecules that actually keep them together. Okay, so this picture illustrates that membrane is what's called a fluid mosaic. Mosaic means it's like there are so many components. So like again, the bulk, the core are phospholipids, but you're gonna add a lot of additional structures. Now, all the structures within the cell membrane, they are not fixed in their position. They move. I think a very good analogy will be the surface of their ocean, right? So you have waves, and you can see this membrane also like kind of like creates this motion. And whatever structures you can find on the cell membrane, they also move. They move with the flow. Okay, so what you can find again, phospholipids, cholesterol molecules that stabilize this whole structure, right? What molecules can sneak in, but still, it doesn't matter because they can't cross, right? So you might find them very, very close to these phospholipids, but it doesn't mean anything, right? You can find carbs. We're gonna talk about different types of the proteins. You find glycolipids, which means fat with glyco, usually referred to carb. So carbohydrates. Glycoproteins means proteins with carbs. Glycolipids means lipids with carbs, right? But again, mosaic model means you have many, many different inclusions, right, within this whole structure. So one thing, just one moment. I'll share the sound. We can actually see this one. It doesn't have sound. It just basically illustrates that, again, you see this, all this red stuff is the water, right? So Jamar, as you can see, yes, water kind of like fills spaces, but still it can't really cross. Let's like watch this again. Yeah, so they move, but they can't, like, you see how
They
many
can't
water
really get
outside?
across.
Yes.
I see there's some in the middle. So like,
Yes, but still, you
very
see,
few. They're
the
stuck.
amount of water in the middle is nothing close to the amount of water outside of
On the
it.
outside, yeah. Okay.
And this is basically about fluidity. And this is basically, you can read about this experiment when they took human amount cell and created kind of like hybrid of this. Okay, not really important, but that's what important here is temperature. Temperature can have an effect. It has an effect on their phospholipids, right? And whatever temperature goes down, it can solidify, right? When temperature goes up, it can actually kind of like expand. To keep it, again, to keep it stable, to prevent this expansion, to prevent its like clumping, that's what you have cholesterol for. So you see, unsaturated versus saturated. So saturated tails will definitely make the membrane more tight, right? Unsaturated will kind of like push. So what we have is literally ideal, one saturated, one unsaturated. They're not too tightly packed. They're not too far apart from each other. And as I said, cholesterol reduces fluidity when temperature is moderate, right? But Whenever temperature goes down, it prevents solidification, okay? So cholesterol literally stabilizes the structure, right? And prevents any fluctuations at temperature changes. That's all you have to know about cholesterol within the membrane. So again, pay attention that cholesterol is not a bad molecule at all, right? Cholesterol is a very useful molecule in human body, in the cells. And cholesterol also plays huge role as being precursor, which means like a template molecule for synthesis of many hormones in humans as well. Any questions here so far? Am I too fast? Guys?
No.
No.
No? Okay, let me know if I'm like, so sometimes I'm talking a bit too much, too fast. Okay, so that's a summary for, again, the ideal structure of the membrane and cholesterol that is very helpful. And we're gonna talk about their proteins. Okay, so proteins are super important part of the cell membrane because proteins are usually hydrophilic. They can interact with whatever soluble with the water. And there are two major types of proteins. Peripheral. Peripheral means that they only on one side of the membrane, either facing outside or facing inside. And integral means that they go through the whole membrane, right? So they penetrate through the hydrophobic core. So another name for integral proteins are transmembrane proteins, okay? So one more time, peripheral on one side, transmembrane goes through the whole entire membrane. And this is transmembrane protein, which is extracellular side, cytoplasmic side. Usually transmembrane proteins, they have in the part that goes through the membrane, they have this alpha helix structure, right? Which basically made of non-polar amino acids. Non-polar amino acids means that they can interact with the hydrophobic tails, okay? So this part is hydrophobic like the tails of phospholipids. So proteins have many functions, variety of functions. And what we're going to talk about, we're going to talk about proteins play a role in transport, which is a very important role. Enzymatic activity means that they can carry out chemical reactions right at the cell membrane region. Signal transduction means they can pick up messages. Cell-cell recognition. In multicellular organisms, they allow immune system to recognize the cell as the part of their whole entire organism. Intercellular joining, basically they help to anchor cells to each other. And they also play part as they also play part of the cytoskeleton, right? You see attachment of the cytoskeleton and extracellular matrix whenever it's needed. That's going to be extension of cytoskeleton outside the cell. Okay, so this is transport. Transport protein, what you can see, can be like a channel, right? So it's literally a channel, but it's a gated channel, which means at a certain point it can close, right? At a certain point it opens. This is a carrier, which means that it picks up at one side and releases on the other side. It's not a channel, right? Because it's usually closed unless it has to move molecules. Enzymes, so you see protein sits on the cell membrane and can conduct chemical reactions right here. Signaling. This is a receptor. Receptor is a sensor. Sensor means molecule can bind, deliver message, right? And message will be like propagated within the cell. Cell-cell recognition. So two cells can communicate. Intercellular joining, keeping two cells together, as a structure. And attachment to cytoskeleton and extracellular matrix. So binding cytoskeleton to structures that are outside the cell. So let's watch this one.
Transport proteins may be carrier proteins that shuttle a molecule, such as glucose, across the membrane by changing shape. Other transport proteins provide channels through which specific molecules or ions, such as sodium, can diffuse. Some transport proteins act as pumps, using the energy of ATP to actively move molecules against their concentration gradient. What other types of functions do membrane proteins perform? Some transfer information. Signaling molecules outside a cell may bind to receptor proteins, which transmit the message into the cell. For example, when insulin binds to receptors on a cell, it signals the cell to take up glucose. Attachment proteins provide support by connecting to the cell's cytoskeleton and to the extracellular matrix. These attachments can also relay information about external and internal conditions. Glycoproteins enable cells to recognize each other. The prefix glycol refers to short chains of sugar molecules extending from the protein. Other membrane proteins can recognize these ID tags. The sorting of cells within a developing embryo relies on such cell-to-cell recognition. Junction proteins form long-lasting connections between cells. For example, anchoring junctions hold muscle cells together. Tight junctions create a leak-proof barrier between the cells lining the intestine. And finally, some membrane proteins are enzymes, which may be arranged in clusters that carry out sequential reactions. In addition to its plasma membrane, a eukaryotic cell is packed with membranes enclosing a variety of organelles. Looking at these diagrams of an animal cell and plant cell, what might you conclude about the importance of membranes in the life of a eukaryotic cell? These internal membranes share many of the functions of the plasma membrane.
Okay, so what you can conclude that they are definitely super important. And for example, it gives you that HIV virus binds to T cell, which is the immune cell, to receptor protein called CD4. So this is special T cells that have CD4 receptor, and there's something called co-receptor CCR5. People basically what happens for the virus to enter the cell, it needs a presence of these two parts, CD4 receptor and the co-receptor. Some people actually have a mutation and they're missing the co-receptor, which actually makes them immune to HIV infection. By the way, HIV stands for what?
Something virus.
Yes. Anyone can tell me what HIV means? Human
immunodeficiency virus.
Very good, Sarah. Yes, human immunodeficiency virus, right? So actually, it was a long road to figure out this co-receptor. And what you can see, co-receptor can be a target for medications, right? So once you block it, it reduces the infection, the viral infection in people who, the person who has the virus, the problem is that this virus infects more and more and more cells. This medication will actually reduce additional viral infection within their T cells. So this is the diagram. This is the receptor. This is the co-receptor. No co-receptor, virus can't penetrate the cell. And the carbohydrates. So carbohydrates, that, as I said, glycolipids, which means, again, glyco, sugar, or carb. Glycoproteins means sugar and proteins. And they usually serve as a markers. Why markers? For the same, again, immune system. Immune cells have to recognize the rest of the organism cells as self, okay? So they usually serve as IDs, like you present your cellular ID, which prevents you from being attacked by immune system. That's crucial for transplantation, right? So when people, for example, need a skin transplantation, kidney transplantation, what else can be like heart transplantation, there is a recipient who receives a transplant and there is a donor. And I'm sure that you heard that there should be a match, right? Match between donor and recipient. The match is pretty much about the immune system. So as long as the immune system is fine with all the structures, the transplant will be successful, okay? If they way different, the transplant can be rejected, okay? That's why transplant usually work the best within the same family, when people genetically close, right? And exhibit pretty much the same, like very similar structures on the cell membrane. Okay, and that's what we're getting to. We're getting to selective permeability, right? We're getting to transport through the cell membrane. Control of traffic. Okay, just give me 1 second. Okay. Okay, before diffusion, we're gonna talk about the water, right? So as I said, molecules that are non-polar, like CO2, oxygen, right? They get, like, they just move through across the membrane, right? They don't really need any assistance. Molecules that are charged, right? Hydrophilic polar molecules, they need assistance. So water, as I said, it's a polar molecule. So water can cross the cell membrane. For the molecules that are polar, there are transport proteins. And I showed you transport proteins could be channels, literally channels that open and close, or can be carriers. Carriers means they open one side and then close and open on another side. What is about water? So water has special channels. I want you to pay attention to this name. The channel for water is called aquaporin. Aquaporin is specific for water. Now, cell can control how much aquaporins are present on the membrane. It depends how much water the cell needs to take in or probably take out, right? So the very busy channels, because you see 3 billion water molecules can pass per second, which is a lot, right? But as I said, water can go through. It can go through the channels. Okay, so I guess this is the aquaporin channels. So what you can see, right? So channels can be present on the cell membrane, right? Or channels could be removed, which means they are on the membrane. And whenever they need it, whenever the cell needs to take the water in, they will be inserted, right? So cell can regulate the water intake. So let's watch this one.
We get all our energy and organic molecules from food. Before we can use the molecules we eat, they have to enter our cells, starting with the cells lining the small intestine. Let's zoom in to the surface of a cell. The plasma membrane is selectively permeable. Some molecules can move across it, while others cannot. How do materials enter and leave cells? Lipids, such as these yellow molecules, can dissolve in the lipid bilayer. Notice how they move down their concentration gradient, from where they are more concentrated to where they are less concentrated. This is an example of diffusion. Diffusion is a form of passive transport. It does not require energy from the cell. Most molecules can't cross the lipid bilayer. Here, the sugar fructose moves into intestinal cells by facilitated diffusion, moving down its concentration gradient through a transport protein. Facilitated diffusion doesn't require energy from the cell, so it's also a form of passive transport. Water crosses the plasma membrane by facilitated diffusion, or by diffusing across the lipid bilayer directly. The diffusion of water across a membrane is called osmosis. The sodium-potassium pump moves ions against their concentration gradient, from where they are less concentrated to where they are more concentrated. This requires energy from the cell and is known as active transport. Energy from ATP is used to move sodium ions out of the cell and potassium ions in. Another type of active transport is co-transport. Here, both sodium ions and glucose move into the cell through a co-transporter protein. Sodium ions move down the concentration gradient created by the sodium-potassium pump, and glucose moves against its concentration gradient. Now let's move to the other side of our intestinal cell. Materials can be exported in vesicles that fuse with the plasma membrane and release their contents outside the cell. This process is called exocytosis. In endocytosis, the plasma membrane pinches in, forming a vesicle that contains material from outside the cell. On this side of the cell, we can also see oxygen and carbon dioxide diffusing across the lipid bilayer. Cells use all these processes to get what we need.
Okay. So for a second, guys, you with me? Yes?
Yes.
Yes.
Okay.
Yeah.
Any
Yes.
questions? Any questions? Any questions so far?
So far, so good.
So far, so good. So let me explain you something. Okay, so you heard that word concentration gradient. So I just want to explain what does it mean, okay? So let's say this is a membrane and you have a lot of molecules, doesn't matter what kind of molecules here, right? And you have maybe like two. You know that there is a constant movement of atoms and molecules, right? You know that molecules don't, how to say, they are not static. They are in motion, right? So what is the direction of the motion? So how do you know which way molecules, substances go? Can you answer that question? what is the direction of the motion, of the movement?
The highest concentration to the lowest.
Exactly, from high concentration to low, right? So the difference in concentration is called a gradient, right? So this is a difference. When the molecules move from high concentration to low, it means along the gradient. Okay, whatever moves along the gradient doesn't require energy, right? Because this is like how it happens in nature. So things move from where you have a lot of them to where you don't have them, or maybe a little bit, right? So naturally, things move along the concentration gradient. Can things move against? No, talk to me. Can substances, molecules move against concentration gradient?
Yes.
Won't they have to be active though?
Exactly. Whenever they have to move against, the energy has to be applied, okay? So no energy is required to move along the gradient, and energy is needed when they move against. Okay, so you can talk about concentration gradient means specifically concentration. It could be also electrical gradient. Electrical gradient means difference in charges. Okay, you have accumulation of charges in one place and no charges, or a little bit in another one. So that's electrical gradient. Okay, so which also plays important role as well in, cellular function. Okay, so the first one is we're gonna talk about passive transport, and the most common one is diffusion, and this is the simplest one, right? So diffusion is movement along the concentration gradient, where you have a lot to where you have a little. So the movement will continue until concentrations become equal, right? Until it's balanced. So here, right, net diffusion, you add molecules of dye, right? You have a membrane. So, and this is the equilibrium, right? So diffusion will continue until equilibrium will be achieved. Can you give an example of diffusion, guys?
Fusion, high concentration, low concentration.
Perfumes.
Like, yes, in real life, like diffusion in real life, please.
Oh, like if you spray something like air freshener and it like spreads throughout the room naturally.
Yeah, or if you apply too much cologne, right?
Oh, yeah.
And people around you will smell it. Yes. What else?
A tea bag in water.
Absolutely, Diwan. Awesome. Or sugar in water, right? Or, for example, my 7-year-old, he never liked milk. He didn't want to drink milk. So the trick was to add food coloring, right? So first thing in the morning, what color do you want this morning, right? It can be green or blue or red or yellow or orange, right? But you add the food coloring, right? And the milk turns another color. So this is
diffusion is the tendency for particles of any kind to spread out from where they are more concentrated to where they are less concentrated. This process can also be described as molecules moving down their concentration gradient. Diffusion across a biological membrane is called passive transport, since the cell expends no energy to move the molecules. Oxygen and carbon dioxide molecules move into and out of cells by passive transport.
And again, carbon dioxide and oxygen, they are non-polar, so which means they don't need any channels or carriers, right? They can cross the membrane the way it is. And yeah, so diffusion can be both ways. usually oxygen goes into the cell and carbon dioxide goes outside the cell, right? So equilibrium can be achieved for both of them. And as I said, it doesn't require energy. 1 second. Hypertonic. I'm sorry, I'm just going through this because I'm looking for, yes. I thought I skipped it because it goes to osmosis right away. I was looking for facilitated diffusion. Okay, so osmosis. Osmosis is kind of similar to diffusion. You can say it's a diffusion of water. So let's say you have a membrane again. So membrane is permeable for water, but it's not permeable for other substances. So let's say you have sugar, you have glucose, right? So you have glucose, glucose. And you have water. This is water. Now, glucose can't cross the membrane, right? Glucose is trapped, but this membrane is permeable to water. The drive to make concentrations equal is very, very strong, right? So what's gonna happen? The water gonna start moving towards glucose. And after a while, what you're gonna see? You're gonna see that level of water here will be higher than here, okay? So this is osmosis. Osmosis is super important thing in, I should say, the human body, or I should say like, yes, in life, huge. Why osmosis is huge? Because if you have any polar molecule, anything that has a charge, right, it will pull the water. Okay, so osmosis, basically, I can say this is movement of water from low concentration of substance to high. Okay, guys, clear so far? Are we good?
Yes, ma'am.
Okay, thank you. So, for example, that's what I was talking about, right? You have selectively permeable membrane, right? So this is the water, something dissolved with the water, and the water will always move towards, again, towards concentration of their solute. Solute, something that is mixed with the water. And there's something called tonicity. Give me just 1 second, guys, just 1 second. Сейчас meeting будет. Guys, 1 second. I just moved to another room, okay?
Okay.
And I'll be right back with you. Okay, I'm back. I wonder, people are gonna go back to offices? Like at some point, it's just crazy, you know. So used people go to work in the morning. People don't stay at home. But
I
I
do
hope
like that.
I hope colleges will like reopen and students will have classes. But I really doubt the companies. I think companies they save so much money keeping the employees at home. So I don't think it's gonna happen. Okay, tonicity. So tonicity corresponds to idea that cells are not, they don't exist in dry environment, right? So cells are within the aqueous solution, right? So remember, outside extracellular fluid has the water, inside intracellular fluid has water. Tonicity means like what is the concentration of solutes in these fluids? I mean, solutes, whatever is mixed with the water. So concentration of solutes outside the cell versus concentration of solutes inside the cell. So that's what is called tonicity, okay? And I'm gonna use the whiteboard again, one more time. So this is the cell, right? And let's say this is extracellular. And this is intracellular. Sorry, no, do it again. Intracellular. And let's say outside the cell, the concentration, if I take specifically sodium chloride, right? So concentration is 0.9%, right? So that's how much salt in the water. You take 0.9 grams of salt and mixed with 100 millimeters of water. So this is concentration of sodium chloride. Inside the cell is the same, right? So if concentrations inside the cell and outside the cell are the same, this solution is called isotonic. So the same. If concentration is the same, right? Inside the cell, outside the cell, the water will be balanced, right? Remember, the water will move from low concentration of substance to high, right? So in isotonic solution, there will be no movement of water in terms of osmosis, right?
Mm-hmm.
You follow?
Yes.
Now, let's say inside the cell is 0.9, but outside the cell is 0. If outside concentration is lower, this solution is called hypotonic. So what's going to happen to the water? Which way the water move? Inside the cell or outside the cell?
Outside.
Why outside?
Oh,
Remember,
because it has a higher water concentration on the inside. I was trying to figure out
yes,
what the percentage were.
well, okay. Remember, water will move always towards high concentration of salt. So the rule of osmosis, whenever you have the salt or anything that is charged, the water will follow, right? So water will follow into the cell, right? Now, if inside the cell is 0.9%, but outside the cell is 9%, this is called hypertonic. So the water gonna move in or out? No, talk to me.
Wait, can you say that again? It got quiet.
Okay, quiet. I'm just saying, if outside the cell concentration is higher than inside, this solution is called hypertonic. Which way the water will move?
Out.
Out.
Yeah.
Out. And the cell will shrink. Okay. So I don't know. So I don't know if you ever heard that. Let's say if you worked out really, really hard, right? And you were sweating a lot. So you lost a lot of sweat, which means you lost a lot of water. Have you heard that you shouldn't really drink water?
No.
No?
I haven't heard that. I don't work out.
Okay, anyone here who works out?
I heard that when you be more active, like you are not supposed to drink water because it makes your stomach hurt.
Yes, but I mean, let's say, again, God forbid, if the person, especially if child, has like stomach flu, right? And the child threw up a lot, diarrhea. What should you give child to drink?
You give a child water because they're lacking fluid.
Not water.
Pedialyte.
That's a lot.
Exactly. Pedialyte.
Electrolytes.
Electrolytes, yes. Why give electrolytes? Just because you give the water, right? The cell will swell. Basically, if the person lost fluids and you give water, I mean, if you're not going to kill the person going to feel very, very sick because all the cells will swell. So after workout, it's better to drink. I'm not really a big fan of all this like synthetic drinks like Gatorade. Gatorade is, in my opinion, it's a chemistry, right? It's just a bunch of chemicals. But a good one is coconut water. Coconut water is much, much better. It has the right amount of electrolytes, okay? So again, you sweating, you throwing up, right? You shouldn't drink just water. You should drink something that has, like, it's better to drink juice because it has electrolytes there, okay? So for example, look at this. This is a red blood cell, right? So our red blood cells, they are inside plasma, right? Plasma is the watery part of the blood. So 0.9 inside, 0.9 outside, balanced, right? Isotonic. The same goes for the like plant cell, right? So you put flowers in the water. Usually it's recommended in some flowers, right? They give you also some like, they give like a bag, right? To add to the water just to make it isotonic. Now, let's say person was bleeding, lost a lot of blood. You want to give the IV, fluids IV. What do you normally give as IV fluid?
Saline?
Saline. Why saline? The same 0.9, isotonic. But let's say you don't have saline. Should you give the person water? IV?
No.
God forbid. Yes, God forbid. You'll kill the person.
Lactic ringers, probably, but not water.
Not of water, right? What's going to happen? The cells will swell and explode. That's it. Death, right? So this is hypertonic. What's going to happen with the water in the plant? The water going to start moving in, right? Moving in, pretty much osmosis is what brings water inside the plant from the roots, okay? The movement of water from, again, from the root of the tree to the very, very top. That's the same osmosis. Let's say you don't have a saline, you want to make it on your own, right? Prepare it by yourself, and you messed up, and instead of 0.9, you gave 9%. What's gonna happen? You see the water gonna move from red blood cells outside. They will shrink. That's the same, like in the opposite, but the same result, right? That's how you're gonna kill the person. And For the plant cell, in a very concentrated solution, the water going to run out and the plant will wilt. That's called plasmolyzed, but it's going to wilt. Okay. So that is,
Senor,
yes.
Professor. Yeah.
Yep. Mm-hmm.
So when a cell is like shriveling up, what is that called again? It starts with the C, right?
Crenation.
Crenation. Yeah. Okay.
Yes, that's the crenation. Yes. So when they shrink. Okay. And osmoregulation is pretty much how organisms control. And one of the ways how we control osmoregulation is urinary system, right? So our urinary system pretty much regulates how many ions should be removed, how much water should be removed. Because again, osmosis is pretty much how body fluids move through different compartments inside the body. That's the osmoregulation. And this is paramecium. Unfortunately, since you don't have a real lab, you didn't have a chance to meet paramecium. It looks like, what does it look like, guys? Doesn't it look like shoe? It looks like a shoe, right?
A shoe? Oh yeah, it does. Okay. I
Yes.
see what you're saying.
Yes, that's how we call it in Russian. We call it a paramecium shoe. I don't know why in English the shoe was dropped. But you see, this is the contractile vacuole. So any excess of water, paramecium will just basically get rid of it, like shoot it out.
The single-celled ciliated eukaryote paramecium is found in fresh water. Water continually enters the cell by osmosis. A specialized organelle in this cell, the contractile vacuole, collects water from the cell and pumps it out with regular contractions. 2 such contractions can be seen in this video clip. Credit, Henry Mainwaring, Western Carolina University.
Okay. And I don't think we're going to talk about this, but also that's like who deal a lot with osmosis are all aquatic organisms, right? So for example, here, those that live in hypersaline, like excessive salty water, they have mechanisms. But if you talk about fish, right? There are fish that live in fresh water. There are fish that live in salt water, like in ocean. Can you take a fish from ocean and put in the fresh water?
No. They'll die.
They will die. Some fish actually can live in both. Salmon can regulate in both environments, the osmosis, but a majority of fish can do that. And within the cell walls, yes, you see, if the solution is hypertonic, the plant will take the water in, right? So whenever you have more concentration inside a cell than out, I'm talking about plant cell, right? So that's what keeps the plants, the leaves, and everything firm. Again, this attraction of water into the plant cells. That's called turgor pressure, and turgid means firm plant. And we going to talk about this to facilitate diffusion, then I'm going to take a break. Okay, so we almost, this is Elodium. This is like a photosynthetic organism. Where's Elodium?
Alodia is a water plant whose thin leaves are only 2 cell layers thick. This 1,000 times magnified view focuses through several adjacent cells in a leaf. In the lower cell, you can see a nucleus with a smaller nucleolus inside it. The huge central vacuole is visible in the middle cell. Chloroplasts can be seen in the stream of cytosol as it sweeps around the cells. Because this is viewed using differential interference contrast, or Nomarski microscopy, the scene appears three-dimensional. Credit, Michael Clayton, University of...
I'm not exactly sure what they wanted to show, but it's okay, Elodium, okay? So if isotonic, the plant isotonic, there will be no net movement of the water in the cell. Okay, so the water will move only in hypertonic. And flaccid, that's what's going to happen to them in isotonic solution, means the plant will wilt. One more time, that's why you have to water plants, just to keep environment hypertonic. And if you put too much salt outside, right, so they will, sorry, if you put too much water, plasmolysis, that's for the red blood cells, the membrane will rupture. And I think that's enough, pretty much. Okay, so it's 9:15, guys. So let's take our 10-minute break, okay? And we talked so far about diffusion. Simple diffusion, simple means it can go through the membrane the way it is. Osmosis is diffusion of water. Facilitated diffusion is diffusion, but not through their phospholipid bilayer, but through carriers or channels, okay? It's like imagine you have, again, imagine you have a room. You have a room and you have a bunch of people outside the room. So they all want to enter, right? But they can't. Why they can't? Because the doors are closed. So you open the door, right? And people move in. So this is facilitated diffusion, okay? Facilitated diffusion, you can't move in unless the door is open. So for this, as I said, channel and carrier proteins. Channel is literally a channel, but it can open and close. Carrier has to change its structure, right? It opens on one side, picks up their substance, and then opens on another side. This doesn't have to change the shape. This one has to change the shape. What is the difference? I believe it's like a nature of the molecules that they move, right?
Proteins embedded in the membrane play a
Can you hear, or I have to do this again? I have to share the sound, guys.
I can hear.
It's shared, it's shared.
key role in selective permeability. For example, this transport protein spans the membrane and provides a doorway through which a particular solute, the red triangles, can diffuse down its concentration gradient. This process is called facilitated diffusion. Facilitated diffusion is a type of passive transport, since the molecules are diffusing through the membrane and the cell expends no energy to move them.
Okay. And one more time, this is a channel protein. And as I said, gated channels, they open when they receive a certain signal, right? Certain channels respond when molecules bind to them. Some respond whenever change in charges, right? Like, for example, in neurons, potassium ion channels will open in response to change in distribution of ions inside and outside the cell. So this is potassium channel protein. You can see this is their ion right here. Basically changes conformation, slightly changes the shape whenever there is a change in voltage. As I said, voltage is distribution of charges. And carrier proteins, as I said, they slightly have to change the shape whenever they move molecule in or out. Good example, glucose. Glucose actually goes inside the cell through the carrier proteins. And that's it about the passive transport. So one more time, passive transport to make concentration of substance equal, right? Diffusion, facilitate diffusion down concentration gradient. Osmosis, the same idea, but when water makes the concentrations even. Active transport is against gradients. Against gradients, it means you have to use energy. And best example of active transport is sodium-potassium pump. Okay, pump is a protein that sits on the cell membrane, right? And this protein uses ATP molecule to move sodium ions outside the cell and potassium ions inside the cell. And this, I want just to explain you something about sodium-potassium pump. Okay. So cells, and if you're gonna take anatomy and physiology, I don't know, like one of you planning to take anatomy and physiology, you're gonna learn about the nervous system, and you're gonna learn about neurons. Neurons are cells that communicate, right? So these are cells for communication. They communicate with each other. They communicate with target cells, specifically with your muscles. And the nature of communication is their electrical current, right? Neurons use like very, very subtle electrical discharges to talk to each other or to talk to your muscles. So these are cells that are able to generate electrical current. I'm not going to go into details how neurons do that, but neurons use sodium and potassium ions to produce the current. So neurons normally keep sodium outside a cell. So you can see the concentration gradient, right? Sodium has high concentration outside. And it's absolutely opposite for potassium, right? It's opposite. You have more potassium inside the cell than outside. But what happens? You still apply rules of diffusion. Sodium will always try to move in, right? Yes? Yes, guys? And potassium will try to move out. Yes. This is what happens when you have concentration gradient, right? So whenever sodium has an opportunity to sneak inside the cell, it will. The same goes for potassium. So neurons are willing to spend energy to keep this distribution, right? So actually, your neurons spend 85% of ATP for operating sodium-potassium pump. It's called sodium-potassium pump. So one more time, what is a pump? It's a protein, right? And one ATP molecule, 3 sodiums out, 2 potassiums in, right? 1 ATP. That's what you can see here, right? Sodium goes out, sodium goes out, ATP is used, and potassium goes in. Again, just to keep distribution against the concentration gradient. That's why it's called active transport.
Sometimes a cell needs to move a solute against its concentration gradient. This process is called active transport, and it requires input of energy from ATP. For instance, most animal cells need to expel sodium ions, Na+, and take in potassium ions, K+, both against their concentration gradients. Here's how the sodium-potassium pump works. Sodium ions bind to a transport protein. ATP transfers a phosphate group to the protein, providing the energy that causes the protein to change shape and move the sodium ions across the membrane, where they are released outside the cell. Potassium ions now bind to the transport protein, and the phosphate group is released. This causes the protein to return to its original shape, releasing the potassium ions inside the cell. The transport protein is now ready to repeat the process.
Yeah, I can tell you, if anything happens to sodium potassium pump, the whole nervous system will shut down.
Watch an amazing video showing how the Na-positive, K-positive ATPase undergoes specific conformational changes as it pumps Na-positive out of the cell and K-positive into the cell. The cytoplasmic side of the cell is shown at the top of the video. Notice how 3 Na-positive ions are pumped out of the cell for every 2 K-positive ions pumped in. Credit: Mark Hilge and Stefan Hilge, Netherlands Cancer Institute, Netherlands.
Why can they say Na, sodium, and K, potassium? Okay. So this is like comparison. One more time. Passive, along gradient, active, against. And there's something that I was talking about, electrochemical gradient. Electrochemical gradient is distribution of charges, right? Difference in charges. This difference in charges corresponds also to something called membrane potential. What is membrane potential? Membrane potential, I don't see a nice diagram, but just in few words, is you remember most of their substances, they have charge, right? It could be iron, or it could be protein that has a charge in it, or it could be, yes, it could be iron or protein with a charge. So distribution of the charges across the membrane is not even. Usually there are a bit more positive charges outside the cell than inside. Okay, so this difference in charges is called membrane voltage. Okay, and membrane voltage, or membrane potential, right? So it's also plays huge role in osmosis, and it plays huge role in, again, for example, for neurons, the way they function. And this is electrochemical gradient that I was talking about as well. And electrogenic pump is a protein that actually, yes, it's basically operates to maintain this voltage difference, right? So this distribution. And as I said, in like mostly it's sodium potassium pump that maintains this electrochemical gradient. And in plants, it's a proton pump. So the same thing is achieved not through sodium potassium, but through hydrogen ions. Okay, so hydrogen ions makes extracellular slightly more positively charged than inside, whatever's inside the cell. Guys, is it clear what I'm trying to say? Talk to me.
This for me is a little bit more difficult to understand, but
I mean, imagine like you have, let's say, imagine you have just, let's say, there's a sodium, right? Sodium, that's it, right? So more sodium outside than inside. That's a chemical gradient, right? Now imagine you have charges. So let's do this way. Sodium, sodium, right? Chemical. But I add charges, right? So now imagine you have more sodium, much more sodium, right? So what happens? Overall, you have more pluses outside than inside. This is electrochemical gradient. Electrochemical, you take into consideration not only concentration of the chemical itself, you also take in consideration the charge. Okay? Make sense? Makes more sense?
yeah, it makes more sense like that.
Yes, potassium also has a positive charge. But the thing is that you have so much less potassium overall than sodium, right? So concentration of sodium is much, much higher. So basically sodium plays more important role keeping outside the cell more positive than potassium itself. Okay, so sorry, than potassium. So overall, outside the cell, you have accumulation of positively charged ions, and you have more of them outside the cell than inside the cell.
Okay.
That's the whole thing. Now, as I said, animal cells achieve this through sodium and potassium. Plant cells, fungi, achieve it the same thing through hydrogen ions that also have a positive charge. Okay, so guys, there are a few more things left, literally. So co-transport. Co-transport is also considered active transport. Why is considered active transport? Because you still use energy to move substances against gradient. But you don't use ATP directly. What you use something that is, you use, it's called a kinetic energy, right? Kinetic energy means the use energy of movement of one substance to move another one. Okay, so try to explain it in an easy way. You see, this is called proton pump. Proton pump is, proton means hydrogen ions, because remember, hydrogen has only one proton. So this is proton pump. You kick out hydrogen, right? And then hydrogen will try to go inside the cell down its gradient, right? Yes, makes sense? Hydrogen will diffuse down its gradient.
Yeah. Passive
So when
transport.
hydrogen will diffuse down its gradient, right, it's going to move. Why wouldn't it take another molecule with it? And it take the molecule against the gradient. So it's like, let's say, I give you an analogy. Let's say you walk into the room, right? You walk into the room that you want to go, and you bring another person. You pull another person with you. So you use energy of your movement to bring somebody else in. That's called the co-transport, okay? This is co-transporter. Co-transporter basically couples movement of substance along its concentration gradient to take another one against. Okay, so like say person doesn't want to move in, right? And you pull the person with you. Make sense? Does it?
Yes, ma'am. Well, to me, yeah.
Yes. So, yes. So, yes, kind of like you want to go and another one doesn't, but you like take by force. This is the co-transport. It's also called symporter. Symporter means they move the same way. Okay, there is antiporter. Antiporter, it's not presented here, but antiporter, you walk in and you take another one out. Okay.
Okay.
This is antiporter.
Is it for hydrogen too? Like, or is it just
not
for?
Necessarily. It's could be it could be other molecules, other substances. I'm not sure exactly which one is used for anti-transport, but again, just pay attention. Co-transport is considered active transport. One moves against along the gradient to take another one against the gradient. And they specifically operate proton pumps to be used to generate concentration gradient for hydrogen. So hydrogen will move in and take something else with it. Okay, so specifically. I think it's really amazing how cells, how intelligent they are, right? Isn't it? Isn't it? Guys, how they know like how to build all the things and operate them?
DNA.
Yeah, I always say intelligence is not a brain. Yes, I agree with you. I think intelligence is a DNA. Like how bacteria know what to do, how viruses know what to do. They don't have brains, but they still know, right? Somehow.
Oh, true.
Yeah. Okay, so we're done with the small molecules. Now we're talking about bulk transport. Bulk transport means large molecules and large quantities. Okay, now bulk transports, they operate through vesicles. Remember, the vesicles, they're bubbles, right, that could be loaded and unloaded. So let's watch the video first and then we'll talk.
Endocytosis is the movement of materials into a cell via vesicles that form from the plasma membrane. Exocytosis is the movement of materials out of a cell by the fusion of vesicles with the plasma membrane. These processes enable patches of membrane to flow back and forth between the interior of the cell and the plasma membrane, reminding us that a cell is a dynamic structure.
Yeah.
Exocytosis is the movement of vesicles in the cell to the plasma membrane, where they fuse with the membrane and release their contents into the surrounding fluid. This process occurs predominantly in secretory cells, such as mucus-producing cells or pancreatic cells that secrete enzymes into the digestive tract. In endocytosis, the plasma membrane invaginates or pinches in, forming a vesicle that moves the enclosed materials inside the cell. There are 3 types of endocytosis, each involving its own specific cell machinery. In phagocytosis, or cellular eating, the cell engulfs debris, bacteria, or other particles by extending pseudopodia around the material. Invagination produces a food vacuole. The material inside the vacuole will be digested after the food vacuole fuses with a lysosome containing hydrolytic enzymes. Phagocytosis occurs in amoebas and in immune cells such as macrophages and neutrophils in mammals. In pinocytosis, or cellular drinking, the cell engulfs extracellular fluid, including molecules such as sugars and proteins. These materials enter the cell inside a vesicle. Epithelial cells and capillaries use pinocytosis to engulf the liquid portion of blood at the capillary surface. The resulting vesicles travel across the capillary cells and release their contents to surrounding tissues, while blood cells remain in the blood. Unlike phagocytosis and pinocytosis, receptor-mediated endocytosis is very specific. It's triggered when membrane receptors bind to specific external molecules, such as protein cholesterol complexes or proteins bound to iron. Membrane vesicles pinch off, and the external receptor and its cargo are brought into the cell.
Okay, so one more time. Large molecules, large quantities, right? So exo means outside the cell, right? And what you can see one more time, Golgi apparatus. Remember, this is an endomembranous system, which means endomembranous vesicle could be created. So let's say freshly made proteins loaded in the vesicle, they move along the microtubules. So the vesicles fuse with the plasma membrane and release their contents. And cells that release hormones or release certain substances, they do this a lot. For example, saliva is released through exocytosis. Endocytosis is opposite. And please pay attention. So this is, for example, amoeba can do the pseudopodium, but not necessarily. Amoeba, any cell, can make this like a fold, right? Load it into the vesicle and then digest. Pino, pay attention, this is for fluids. Fluids with, let's say, fluids with something, right? Again, engulfs, takes it in. Phagocytosis, this is for solids. That's how immune cells literally eat germs, right? They engulf them, take them in, and digest, break them down. Pinocytosis, like how certain, again, single cell organisms drink, take water in. Receptor-mediated endocytosis is more specific, right? So you have a big delivery. This delivery means substances have to bind to receptors, they have to be recognized, and then the vesicle will be formed and the contents will be taken in. Okay, so everything will be digested and a delivery will be available. So one more time, phago basically means to eat. So phagocytes, cells that can eat other cells. Again, our immune cells. Big group of them are phagocytes. Amoeba does phagocytosis. It eats algae cell, right? Whatever is phagocytosis. Pinocytosis, as I said, is a fluid. Fluid with something, right? Pinocytosis. And this is the animation.
In pinocytosis, or cell drinking, the cell engulfs extracellular fluid, including molecules such as sugars and proteins. These materials enter the cell inside a vesicle. Epithelial cells and capillaries use pinocytosis to engulf the liquid portion of blood at the capillary surface.
I think we watched this, right? And receptor mediated, as I said, binding, right? Binding to receptor, coating this vesicle with a specific solutes, right? And then taking it in and digesting. Let me just see. Guys, we're almost done. Wow. Yeah, like receptor-mediated endocytosis is actually to take cholesterol. That's I also wanted to tell you. And but you remember, cholesterol is needed for being a part of the cell membrane. And this is the last animation, and we'll be done, and I'll take the attendance.
We get all our energy and organic molecules from food. Before we can use the molecules we eat, they have to enter our cells, starting with the cells lining the small intestine. Let's zoom in to the surface of a cell. The plasma membrane is selectively permeable, called osmosis.
Come on, we watched this. Okay, sorry. I don't know why they put it at the beginning, but we watched this, right? We did at the beginning. Guys, anyways, like this is your I think I posted for you in PDF.